Greetings, viewers. I want to welcome you to another YouTube class. In today's class, we're going to be talking about Fasciola SP. Fasciola SP is one of the parasites found within the fluke and within Trematoda. I've thought about the first category or the first type, the first example of these parasites, and I mean parasites, sorry, that are found under Trematoda, and that is Schistosoma, and that is why I want to use this time to talk about Fasciola. And Fasciola has two main species, the Hepatica and the Gigantica. Hepatica and Gigantica. And it's a parasite of plant-eating vertebrates. Plant-eating vertebrates. Vertebrates that eat plants. They are the group of animals that are usually implicated in the disease called fascioliasis caused by this parasite that is called Fasciola sp. Now, there are basic differences between fasciola and schistosoma, even though they are closely related, and of course they are found in the same class Trematoda, but there are basic differences. Number one difference is that in the case of fasciola, in the case of fasciola, two intermediate hoods are involved: the snail and the watercress. But in the case of schistosoma, only one intermediate host, and that is a snail. Another difference is that the infective stage of fasciola is called the metasaccharia, whereas the infective stage of schistosoma is the saccharia. Now, the last time we talked about schistosoma, I said that the infective stage is bifid, meaning that it has two extensions within the tail. This is how it looks like. But this is not how the infective stage of fasciola looks like. The infective stage of fasciola is it looks like a spherical object. It's spherical, and this is a saccharia. Fasciola still has saccharia, but the saccharia is not the infective stage. So in this case, it is when the saccharia leaves the snail intermediate tools and now migrates into a watercress. That is where it now forms the infective stage, which is now called the metasaccharia. So that is another difference between fasciola and schistosoma. Then, of course, you should also note that even though they use snail as the intermediate host, but the species of snails are not the same. For schistosoma, I mentioned the Bionfalaria, I mentioned Oncomelania, and I mentioned the Bulinus globosus, and that is for Mansoni, for Japonicum, and for Hematobium. But in the case of fasciola, the case is different. The name of the snail intermediate host is Lemni natalensis. Then when it comes to similarities, of course, they are similar. Don't forget they belong to the same class Trematoda. They use the same aquatic environment for the propagation of their life cycle. And of course, they use snail as their intermediate host. So these are some of the similarities between fasciola and schistosoma. Quickly, let's go into the life cycle of fasciola. Coming to the life cycle of fasciola, it begins by the ingestion of metasaccharia. Okay, this is another difference sorry, between fasciola and schistosoma. In the case of schistosoma, the life cycle, method of transmission, is active in the sense that the saccharia, which is the infective stage of schistosoma, actively penetrates through the unbroken skin of the individual that is swimming within water. But in the case of fasciola, it involves the individual going to pick the watercress within the bank of the water body and ingesting it. It is only when the person ingests it that the person will now ingest the infective stage of the parasite, which is now called the metasaccharia. So when the person ingests the infective stage of the metasaccharia, that metasaccharia is just like a cyst, and by the time it undergoes what we call excistation, and I mentioned this excistation method in entamoeba isoretica. So excistation means the breaking of the cyst, and once the cyst is broken, then you will now discover that the fluke will now be released into the intestine. And in this case, the part of the intestine is within the small intestine, the duodenum. So when a station occurs, the fluke is released, that's the fasciolar parasite. And of course, once it's released, it now migrates through the duodenal wall into the peritoneal cavity. From the peritoneal cavity, it penetrates the capsule of the liver, and of course, ultimately, it settles within the biliary tract, transversing the parenchyma before it now settles within the biliary tract. Now, this biliary tract is also called the bile duct. The bile duct. If you look at the shape of the liver, the shape of the liver, the shape of the liver, it's covered that there we have channels. Channels. This is the gallbladder. We have channel through which the bile flows. Now that tract through which the bile flows or the bile is excreted, you know, and of course I wouldn't want to go into the function of bile. Of course, we should know it's used for emulsification of fat. Then that channel through which the bile flows into the intestine is what we call the biliary duct, and that is where this parasite goes to mature into adult. By the time it matures into adult, of course, it begins to lay eggs. And when the individual passes out physis, then the eggs or the parasites are released into the external environment. By the time they are released into the external environment, they hatch into what we call the meracidia. And once the meracidia hatch from the eggs, of course, they look for a snail host to penetrate. By the time they see an appropriate snail host, the parasite must make contact with the appropriate host at the appropriate time before a parasitic host relationship can be established. So if the parasite does not make contact with the appropriate host at the appropriate time, then a parasitic host relationship cannot be established. So the appropriate snail host for fasciola is the limna natalensis, which I've said before. So once this miracidia, encounters the species limna natalensis, then of course it penetrates into it, and by the time it penetrates into the snail host from the miracidia, it metamorphoses into the sporosis, and for the sporosis, metamorphoses into the radii, and for the radii to Saccharia. Once you form Saccharia, don't forget that Saccharia is still not the infective stage. From this Saccharia, it leaves the snail into sorry, sorry, it leaves the snail intermediate host looking for a watercress or a grass, those grasses within the water bank to encrest itself. Once it sees one, then of course it goes there to increase itself, and that is the end of that cycle. Then when an individual goes to this water body to get those vegetables and probably eat them without properly cooking, then that is how the cycle continues. So this is a kind of description of the life cycle of this parasite. Then, of course, apart from this, going further to talk about the pathology, what does this parasite cause? So the person may experience urticaria or what we call hives, especially that location, especially on the skin of the individual. The person may experience what we call the urticaria, 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 or hives. Hives. This is a kind of a hypersensitive reaction of the body. Sometimes you see it, you see a kind of reddish bumps on the body, on the skin of such an individual. That is what we call the urticaria. Then the person may, of course, have slight fever or inflammation of the liver, and the spleen and the liver can be inflamed together. The inflammation, the inflammation of the liver is called hepatomegaly. Hepatomegaly. The inflammation of the spleen is called splenomegaly. But sometimes we can use one word to combine these two: hepatosplenomegaly. megaly. This is to show the inflammation of the liver and the spleen at the same time. Then, of course, another effect for this parasite is the fact that the person may have increased level of eosinophil. Eosinophil is a type of a white blood cell that is usually implicated in parasitic infections. And once there is an increased level of this eosinophil, it is now called eosinophilia. That condition is called eosi- eosinophilia. Eosinophilia. That is increased level of the type of white blood cell that is called eosinophil, and is not a good one. Once there is eosinophilia, it can result to multi-organ damage and other effects that are not too good to the body. So these are some of the pathological effects of this parasite, this disease, sorry, called fascioliasis. The name of the disease is called fascioliasis, as I've mentioned before. How can we prevent and control this disease? Of course, watercress or vegetables within water bodies should be properly cooked before they are taken, and we can also destroy the mollusk or the snail using a molluscide. Then, of course, treatments. What kind of drug can be used in the treatment of this parasite? You can use triclabendazole, triclabendazole, triclabendazole can be used. Then biotinol, biotinol. These are some of the drugs that can be used in the treatment of fascioliasis. This is where I will stop the class for today. In our next class, we'll be, talking about another category of pretermitis, which is now the cystoda. We are done with the trematoda. So under cystoda, we will now be looking at the life cycle of the tinea cilium and tinea saginata. And as time permits, we may look at Echinococcus granulosus, Haemolepis nana, then maybe Diphyllobotum latum and Spirometra. Thank you once more for listening. See you in my next class.